So very good evening to the student who have joined. Can you let me know in the chat box if my voice is audible to you, and also if you can see the PPT on the presentation. Let me know. Yeah. So everything is fine, right? The voice and also the PPT. We will wait till the students join and then we will start today's session. Hello, let us wait till more students join and then we can start our session. Meanwhile, let me know if my voice is audible to you and if you can see the presentation. You can use the chat box to communicate with me. We will start the session soon, so please wait. I hope my voice is audible to you and the presentation is also visible. You can use the chat box to communicate with me. We will wait till it is 6 p.m. and then we will start today's session. I hope my voice is audible to all. If not, you can write in the chat box if there is any problem by hearing my voice or seeing the PPT. We will wait till more students join and then we can start our session. So yeah, if I want to clarify a few things before starting the sessions. For having a smooth streaming of the class, I have muted you all, and also the chat box is open here. So if you want to tell me anything, any question, any queries, anything, you can just write in the chat box. And firstly, I hope my voice is audible to all. Can one or two people write in the chat box if my voice is audible to you? And also if you can see the presentation. Yeah, thank you so much. uh, we will start soon, so please wait. We will start in 1 or 2 minutes. Let other people join too, and at sharp 6 p.m. we will start today's session. So I am recording this session. So in case anybody has any network issue or anything, so don't worry, because since I am recording the session, so I will upload it in YouTube, and the link will be shared to you by NPTEL. So you don't have to worry about missing anything due to poor network. That's why I have muted you all, so that I can record the class smoothly. And if you have anything you want to tell me, any query, any questions, anything, if you have problem seeing the PPT or my hearing my voice, everything you can write in the chat box. Okay, so it is 6 p.m. I think we can wait for 1 or 2 more minutes, or we can start the sessions also. Let a few more people join, then we will start today's session. So now let me know if the presentation is visible to all. If the PPT is visible to all, please let me know in the chat box. Okay, thank you so much. Since I have told you that the chat box is open to all, and I'm watching the chat box every time, so if you have anything you want to tell me, any query, any question, you can write in the chat box. So let us start with the introductions. So very good evening to all. My name is Mousumi Deb, and I am the PMRF fellow from the Department of Chemistry and Chemical Biology, IIT ISM DHANBAD. I will be your TA for this NPTEL course of Medicinal Chemistry, this NOC24-cy17. For this course, I will be the TA in the problem solving session. So, I am a PhD student from the Department of Chemistry, Chemical Biology, IIT ISM DHANBAD. And we will have this kind of problem solving sessions for the next 12 weeks, because you have 12 week. This is a 12-week course. So right now there is a second week is going on, right? So today's class we will discuss week 1 and week 2 together, and from next day onward we will move to the week 3, week 4, and so on. So every week we will have this kind of problem solving session on Tuesdays. Okay, so since I am recording the session, and if there is any problem, you want to tell me anything, you can write in the chat box, because for smooth streaming and smooth recording I have muted you all, but the chat box is open always, so you can let me know anything. Okay, if you have any problem, any query, anything, you can let me know in the chat box. So before starting, I will point out few things. So there will be this kind of problem solving sessions every week. Okay, and these sessions will be solely on Tuesday from 6 p.m. to 8 p.m. So please make it convenient to attend the classes, because these sessions will be live on Google Meet platform. Although I will be uploading the video, the recording of the class later, but still I think this kind of interactions are more preferable, because I can then know your what is your query, what is your problem, so we can discuss it in the next class also. So all these sessions will be live on Tuesday from 6 p.m. to 8 p.m. on Google Meet platform. The link will be same. The link which you have used to join today's class, the link will be same for next 12 weeks. Okay, and after 12 week we will also have a 1 hour of doubt clearing session or summary session kind of thing, so before examination. So you don't have to worry if you have any query which is not clear during the all the 12 weeks. You will have one more 1 hour class to clarify your doubt. Okay, so you don't have to worry about it. So these sessions, during these sessions, as I have told you that for smooth streaming I have muted you all, but still you can write anything, your questions, your doubts, your query in the chat box. Okay, at the end of the session I also will share my email ID to you, so that if you have any problem after the class also, you can let me know via email. Okay, I will try to clear your doubt in the next class. So I will share it at the end of the sessions. So in this session, okay, one more thing, this all the sessions it will be since this is a Google Meet platform and everything is and there may be a network issue or network disruption happen anytime. So when a network disruption happens, so I may there is a problem, so please do not leave the class immediately. Okay, if the problem is from my side, so please wait for some time, I will rejoin soon. So but don't leave the class in between, just wait for some time, I will make sure to join as soon as possible. And if there is a network issue from your side, you can just rejoin anytime in between the class. Okay, so there is no problem in that, but if there is a network problem in my side, so please wait for some time. So during the class, you can just write your doubts query throughout in the chat box, and if there is any doubt and query persist, as I have said, I will share my email ID and you can just drop the email ID in that email. And also, if you have any doubt after that also, there is a discussion forum in the NPTEL SWAM website. You can write your doubt or query at that portion. Okay. And this presentation, whatever I am telling you today, this presentation, this PPT and this class recording, it will be shared to you soon by the NPTEL, so you don't have to worry about anything. So I think we can start today's session. So what we will do in this problem solving session is we will solve few questions. Okay, so right now you are learning the medicinal chemistry, and this is the second week. I think 2 weeks are live right now. There is a the videos and recordings and everything materials have been shared to you. And this today's class, the motto of today's class is to help you out with the assignments. Okay, because every week you have a weekly assignment, right, which you have to solve and you have to submit, and you have marks on that weekly assignment. So how you have to prepare yourself for this kind of assignments. We will solve questions in this problem solving session. So I will present you 20 questions today. Okay, and this 20 questions will be from this week 1 and week 2. Since 2 weeks are right now live, so we will have 20 questions from week 1 and week 2 combined, and these are all are MCQ questions because your assignments are in MCQ format, but your final examination, I think it will be in descriptive format, so we will talk about that later on. So right now we will solve, I will show you 20 MCQ questions. After I show you one question, I want all of you to try to answer that question. Okay, there will be 4 options, A, B, C and D. All of you try to answer that question in the chat box. In the chat box, write which of the following you think is the correct one. There is no marks given for today's session. Okay, there will be no marks, so you don't have to worry about being wrong or anything. But you have to answer the questions. That's how I will understand if you are understanding the course or not, and if you have any problem, I can help you out. Okay, so please try to answer these questions. So as I have said, today it is the there will be little bit, so yeah, let other people join, we can just add them up meanwhile. So as I have said, today we will solve questions from week 1 and week 2 combined. Okay, now because you have to submit your week 1 and week 2 assignments on 7th of February. Okay, don't try to submit your assignments timely. Okay, submit your assignment timely, don't miss the deadlines, it will be better for you. So what is in the week 1 and week 2 for this course of medicinal chemistry? So in week 1 we have introduction to medicinal chemistry part 1 and part 2. This is in two part, the introduction of medicinal chemistry is given. Then we have intermolecular binding forces. Then we have this protein structure and function. Then after that we will have tutorial 1, which is on acidity, basicity and the related concepts. Then tutorial 2 will be on basic concepts of thermodynamics and kinetics. After that, in week 2 we will have enzyme catalysis part 1 and enzyme catalysis part 2. And week 3, tutorial 3 have this binding forces, protein structure and function. Then we have this introduction to receptors. After introduction we have receptor types and functions, and tutorial 4 finally on receptors, binding interactions and ion channels. So now right now I will just make it a presentation mode, and I will show you, or I will present you the questions, and I want all of you to try to answer those questions. Okay, there will be 4 options, A, B, C and D, and you have to try to answer the questions. You may be, it may be wrong. If even if it is wrong, please still try to answer. So the first question of today's session is, what is meant by a lead compound in medicinal chemistry? Your options are: number A drug containing the element lead. Number B, a leading drug in a particular area of medicine. Number C, a compound that acts as the starting point for drug design and development. And number D, a drug which is first to be prescribed for a patient for a particular ailment. So what is meant by a lead compound in medicinal chemistry? After you try to solve the question, after I see few answers in the chat box, then I will show you what is the correct answer, and we will discuss this particular topic. For each problem we will do it in the same way. We will I will show you the question and the options. You will try to answer the questions, and then we will discuss that particular topic. So what is meant by a lead compound in medicinal chemistry? Your options are: number A drug containing the element lead. Number B, a leading drug in a particular area of medicine. Number C, a compound that acts as the starting point for drug design and development. Number D, a drug which is first to be prescribed for a particular ailment. All of you, please try to answer this question. Even if your answer is wrong, there is no marks given for the sessions, so you don't have to worry about it, but all of you must try. That's how I will understand that if you have any problem understanding a particular topic or not. What is meant by a lead compound in medicinal chemistry? I will give you some time, so think and then give the answer. Let me check the chat box if you have. I have few answers by now. Okay, so I think I have few answers in the chat box. So most of the answers are telling that it is the option number C. Okay, now let us check what is the correct answer. And yes, this is the very good response. Thank you all for very good response in the chat box. For all question, I want all of you to try to answer the questions. Okay, so the most of the answers are given that number C, a compound that acts as the starting point for drug design and development. It is the correct one. So let us check what is the correct answer. So the correct answer is very good. It is number C, a compound that acts as a starting point for the drug design and development. So now let us discuss. So what is a lead compound? So medicinal chemistry, basically a chemistry where we just find a correlation between a particular compound with its medicinal property or its therapeutic effects. Okay, so in this particular medicinal chemistry, lead compounds are those compounds which demonstrates a property likely to be therapeutically useful. The level of the activity and the target selectivity are not crucial for the lead compound. It is used as a starting point for the drug design and development, and it is found by the design like molecular modeling or NMR or by screening the compounds. So we have to need to identify the suitable test in order to find a lead compound. And what is the active principle for it? A compound that is isolated from a natural extract and which is principally responsible for extract pharmacological activity, often used as a lead compound. So what is a lead compound? So whenever there is a drug design or development process, first we discover what is the target. Okay, what is the particular target for the drug design or development? Based on the target, we have to find a starting compound or a starting point which have some therapeutic effect on that particular drug target. Okay, this can be a natural product, this can be an existing molecule in the database, or this can be a newly designed molecule. After we identify this particular lead compound, then we have to go through a particular series of a cycle particular, which is a drug discovery or development cycle. Okay, and for this drug development, okay, if you want to tell anything, or if you want to say anything, you can just write in the chat box. Okay, if you have any doubt, any query, you can write in the chat box. So how this drug discovery circle works. Now first we have to identify a starting point or a lead compound. Okay, and how we will this drug discovery cycle works, you can see from this flow chart. So the first is a compound collection. So we have a collection of compound. It can be a pre-existing compound, or they can be a series of newly synthesized molecule, or it can be a natural product database also. So first we have to go through some primary assays or high throughput assays or in vitro studies. So what is all this about, we will slowly learn. Okay, so basically we have to screen the compounds. First there is a primary assays like high throughput screening or in vitro screening. Then we have secondary assays like your counter screens, viability, toxicity, metabolism. All these terms, all these things we will learn slowly or gradually. Okay, no, from this data collection from some primary and secondary assays, we identify a lead compounds, and after identify the lead compounds, we perform SAR. What is SAR? SAR is a structure activity relationship studies. Okay, after this SAR perform, we have the structural characterization of the protein ligand complex, which is a direct way to design a compound, or we can have this indirect way, which is a chemical throughout the chemical synthesis, and then this cycle repeats until and unless we have a very good compound which can be used as a clinical candidate. Okay, so we'll always remember the starting point for a drug discovery or development process is finding a lead compound. We have some biological activity or some therapeutic effect on a particular drug target for which we are trying to find the particular drug. Okay, gradually we will learn all about all this things like what is a counter screen, what is toxicity, biology, viability. We will slowly we will learn that. Okay, so now the second question is the correct order of a drug discovery program is number A or number 1, there is a target discovery, number 2 lead identification, number 3 lead optimization, number 4 hit identification, number 5 preclinical development. So you have to identify what is the correct order of these 5 things. Okay, number A, option A is 1 4 2 3 5, option B is 2 1 3 4 5, option C is 3 4 1 2 5, and option D is 1 3 4 2 5. Carefully read all the 1 2 3 4 and 5, and then try to identify what is the correct order of the drug discovery program. Just like the last question, I want all of you again to participate and write in the chat box whatever you think is the correct order of this drug discovery program. Again I am saying I have muted you all, and so that we have the smooth streaming of the class, and also I can record the class smoothly. But if you have any doubt, any query at any point of class, the chat box is always open and I am monitoring the chat box. You can just write it in the chat box. So what is the correct answer, or what is the correct order of a drug discovery program? Take 2 minutes and then try to answer this. Let me check once if there is any student who want to join. Okay, I have few answers, I think. Okay, I think number A, number B, number A. Okay, so I have few number A and few number B. Okay, let me check. Take few more minute. Others also try. What is the number A till now? Number A and number B. I have these two. I have seen these two answers in the chat box. Others also try. What is the correct order of a drug discovery program? Target discovery, lead identification, lead optimization, hit identification, and preclinical development. Which comes first and which comes later? Somebody wants to join. Okay, so let us see what is the correct answer for problem number 2. I can see that most of the answers are about number A, or I have few number B also. So let me check now. So the correct answer will be number A. First, there is a target discovery, then we have hit identification, then we have lead identification, lead optimization and preclinical development. So how this particular drug discovery program goes through? So first, what is the first thing about when a medicinal chemistry program or a drug discovery program? First, we have to identify a target discovery or validation. So what I mean by a target discovery or validation, for a particular disease, to tackle a particular disease, there are many ways. Okay, just for example, let's say blood pressure. Okay, so to minimize the blood pressure, there are various targets. Okay, there are 3 to 4 targets or 3 to 4 way by which we can minimize a patient's blood pressure. This is same as for every disease. Okay, may it and bacterial causing disease or a viral infections or a cancer or this kind of ailments, everything we have multiple targets. Okay, so first we have to validate if the target which are choosing for a particular disease, is it valid enough to start our medicinal chemistry program or drug discovery program. Okay, so first we have to identify what is the target IIT or target prioritization, disease target association and ligandability assessment. Okay, what is the ligandability assessment? Since we are deciding a target, we have to find out if we can tackle that particular target. Okay, if we can develop a compound which can interact with the particular target, first we have to validate these things before starting the next, before moving on to the drug discovery program. So what is the second thing? Second thing is the hit identification. So what is the hit identification? Hit identification, by the methods of hit identifications are the protein modeling, virtual screening and library enumeration. One second. Okay. So what is the mode of the hit identification? Is the protein modeling, then we have virtual screening and library enumeration. Means we have to model the protein. If you are the particular target or the particular cell, let's say you have a find out a disease on which you want to work on. Okay, and that particular disease can be tackled by inhibiting a particular enzyme, then that particular, or activating a particular enzyme, then that particular enzyme or that particular protein is your drug target. Now after identifying the particular target, you have to perform the protein modeling, okay, of that particular protein or particular target to check what is the active site which have to activate it or which have to inhibit it to have this particular medicinal or therapeutic effect of your compound. So that is called the hit identification. Then we have the virtual screening. We have to screen all the compound libraries for which are present to tackle or which interacts with the particular protein, and from which we have a compounds or few compounds which are the hit compounds for this particular target. Then we have to identify the lead. As I have told earlier, what is a lead compound? It is a starting point in your medicinal chemistry or drug discovery program. So it can be done by few modes, like we can have a scaffold hopping, we have binding mode predictions, and these all are we will learn slowly. Okay, we can have this physical chemistry property prediction, we have this synthesis planning and in silico admit property. What is in silico? In silico is mainly basically the computational methods. Okay, in silico means it is the computational method, the methods which can be done by using softwares or the computers. Okay, and what is admit we will learn gradually or later today in today's class. Then we have to optimize the particular lead or lead optimization, which is the multi-parameter optimization, which is the prediction of DMPK endpoints, generative modeling, PKPD, PPK modeling. All this term we will gradually learn slowly. Like PKPD is a pharmacokinetic pharmacodynamics, which I think you have heard before, but you don't know what is exactly. So we will learn it slowly. And then we have this preclinical development, where we have this dose prediction and biomarker discovery and everything. So this is a very shortened and very broad, I mean in broad sense, this is a drug discovery program. We have few very short things in this particular drug discovery pathway. So this is a very summarized way to see it. So these are the correct order was the target discovery or validation, hit identification, lead identification, lead optimization and preclinical development. So now we will move forward. So let's see what is the problem number 3. So problem number 3 says, when a drug is approved for human testing, so when a drug is approved for the human testing during clinical trials, your options are number A, between phase 3 and phase 4, number B, between phase 1 and phase 2, number C, at the end of phase 4, and number D, at the end of preclinical trials. So identify which one of the following is the correct answer and let me know in the chat box. In last two questions you have responded very well and most of your answers were correct. So now I want all of you to again try for this question. The problem number 3, it says, when a drug is approved for human testing during clinical trials. Your options are number A, between phase 3 and phase 4, number B, between phase 1 and phase 2, number C, at the end of phase 4, number D, at the end of preclinical trials. After you try to answer the question, I will show you what is the correct answer and we will discuss what are the phase 1, 2, 3 and 4 for this particular drug discovery program. Okay, I have few answers. Problem number 3, I think. So number D, number D, number D. So many of you have said it is number D. Okay, let us wait for 1 or 2 more minutes and let others try and then we will answer these questions. When a drug is approved for human testing during clinical trials. Let me check again if I have any other options. Okay, I have few number C also, number A. Okay, nice. So we have many options in this question, which is good. We have various answers. Okay, let us check and then let us discuss what are the phases of the drug discovery program or what is clinical trial. In the last slide, what I have shown you, it is up to the preclinical development. So it is one part of this drug discovery program and this part is basically the medicinal chemistry programs work in this particular till the preclinical developments. After that, what happens? After that, the clinical trials begin. What are the clinical trials? And there is few phases of this clinical trials for a particular new drug to be approved for use. So the correct answer, let us see first what is the correct answer. So the correct answer is number D. At the end of preclinical trials, a particular drug is first approved for human testing. Okay. So what are the clinical trials? So this is a clinical trial process. So first, in the last slide, what I have shown you, it is up to the preclinical trial, right? We have a particular target for a particular disease. Then we have this hit identification, lead modification, preclinical studies. After the preclinical studies, we have this clinical trial process. Okay, and this clinical trial process takes up to 5 to 15 years. Okay, for a new drug to be approved for particular disease. But these special designations can speed up the process, like we have seen during the COVID vaccine, right? So the vaccine was identified and everything was done in so fast time. So this clinical trial process can be speeded up if the special requirements happen, like this kind of pandemic disaster. Or otherwise it takes nearly 5 to 15 years for a new drug to approved for a market surveillance. Okay. So first we have this preclinical studies where we explore if and how a new drug may work. The drugs which are approved after the preclinical studies can be now tested on the humans. Okay. So first the human testing begins at the end of the preclinical studies. Then we have few phases of this particular clinical process. The first is a phase 1. Here we explore the safety and the dosing of the drug. Then we have phase 2 where the exploration of the effectiveness of the drug is done. Okay. In the phase 3 we have the exploring the safety and effectiveness compared to currently available treatment. So the phase 3 is all about the differences between already existing drugs and the new drug. Okay. And then we have, after this phase 3, we have to submit the drug for a FDA approval. FDA is the Food and Drug Administration. Okay, you have heard FDA. I think if you see there is a drugs at the medicines you are taking, it will be FDA approved. Okay, after FDA approved, the FDA reviews the particular drug. It is an organization. We will learn about these organizations at the end of the your course. Okay, what are the organizations which approve the drugs for a particular use? And this FDA, what they do, they confirm the safety and effectiveness of the drug. And then after FDA review, if the compound or new drug passes this review, this drug is approved. Then we have the one phase 4. Okay, and this phase 4, what happens? The evaluation of the drug over time in the large number of patients. Because the clinical trial process, the clinical trial process happens in a very small number of patients, right? But when once the drug is approved and it is used in a particular population of humans, then the effect can vary to person, and this phase 4 that evaluates if the drug is still effective over time or it is effective for a larger population of the patients. Okay, so that this is a particular drug discovery or clinical trial process for a new drug to be administered. Okay, so I think it is clear. So till now, if you have anything, any problem, just let me know in the chat box, or any problem you have listening to my voice or presentation, seeing the presentation, you can just tell me. Okay, so very good, most of you have identified the correct answer. So now the next problem. So problem number 4 says, which of the following areas is not a part of preclinical trials during drug discovery? Your options are: number A, drug metabolism, number B, pharmacology, number C, toxicology, and number D, structure activity relationship. Again, try to answer the question in the chat box. Which of the following A, B, C or D option you think is the correct one for this particular problem number 4, which says which of the following areas is not a part of preclinical trials during drug discovery. I will give you 2 minutes. Read the question carefully and then try to answer it. Okay, somebody wants to join. Okay, so I have few answers, number D. Okay, I have few number D's. Let others also try. I will give you 1 more minute. Try to answer. Even if your answer is wrong, it doesn't matter, but please try. Okay, I have one number C, then number D again. Okay, let us check what is the correct answer and then we will discuss it. Okay. So, the correct answer will be yes, number D, structure activity relationship or SAR is not a part of preclinical trials during drug discovery. So, what is the exactly what happens during the preclinical trial or testing? So, a laboratory test for a new drug or a new medical device usually done on animal subjects, okay, to see if the hoped for the treatment really works and if it is safe to test on humans. Many preclinical trials include pharmacokinetic study on how the drugs move through the living organisms. The 4 processes are examined in pharmacokinetic studies are absorption, distribution, metabolism and excretion. The other tests includes chemistry tests like purity, stability, shelf life of the product, as well as the development like dosing, packaging, administration, etc. So basically what happens during the preclinical trial or testing, so in the, what happens that the new drug or new compound, it is tested on animals. Okay, so there are very animal subjects during a drug discovery program, and very common is the rat model or mouse model we say, because why mouse are used for the preclinical trials, because these have most of the similarities with this human system, or the proteins and everything, the DNA, and they have the most similarities. That's how this laboratory in mouse or any other animals are happen. So there are many models. Okay, not necessarily always the on the particular organism it is tested. It can be tested on a models also, which is a computational models also can be happen. So during the preclinical study, we have this drug metabolism, pharmacology, toxicology studies, how this drug is administered or absorbed, distributed and metabolized. But SAR or structural activity relationship thing is, it is a different thing. Okay, so what happens during the SAR particularly, we have to identify that, so a particular drug or a particular compound, there are various functional groups. Okay, so let's say there is an organic molecule and it has let me just draw it out. So let's say you have a particular core, organic core. Okay, let's say it is a benzene. Okay, it is a hypothetical molecule, let's say it is a hypothetical drug, and we have this this is the structure is, there is R1, and for just forgive me for this bad handwriting, because it is a little bit difficult to control it here. Then let's see, we have this here, we have this R2, and let's say we have this R3. So what happens during basically the structure activity relationship studies, we have first we try to keep one group where we try to keep two groups same, and just we change one group. So first we will have, if when we have, let's say it is my lead molecule. Okay, okay, so let's say this is my lead molecule, and after identifying the lead molecule which have a few therapeutic effect for this particular disease, we have to modify this lead molecule. So how the modification will happen, we will use this structure activity relationship or SAR. So what happens, so if it is a lead molecule, first we keep these two positions unchanged and we change this R1 group. After having a better compound or better activity, we then keep then we will keep R1 R2 or R1 R3 same and change R2, and then similarly we change R3. That's how one by one the groups are changed, and we have to see if the activity changes. Okay, so that's how the structure activity relationship studies perform, but it is not a part of a preclinical studies. Okay, basically it is a part of a lead optimization. The structure activity relationship is a part of a lead optimization or lead modification. Okay, but in the preclinical study, what happens, the particular drug is tested on the particular animals before trying it on the humans, to see how this drug is absorbed, distributed, metabolized and excreted, and also the purity, stability, shelf life of the drug, and the dosing, packaging, administration etc. also are tested during the preclinical trial. Okay, so moving forward, the problem number 5 says SAR shows that the important binding groups in morphine are phenol, aromatic ring and amine. A diagram can be drawn which shows that these functional groups and the relative orientation. What is the term for this particular diagram? Read the question carefully. Okay, the SAR or structural activity relationship shows that the important binding groups in morphine are phenol, aromatic ring and amine. A diagram can be drawn which shows these functional groups and their relative orientation. What is the term used for this particular diagram? Your options are number A, pharmacodynamics, number B, pharmacokinetics, number C, chromophore and number D, pharmacophore. Again, I want all of you to try to answer the question in the chat box. Even if your answer is wrong, it doesn't matter, but please all of you try to answer this question in the chat box. A, B, C or D, which of the following options you think is the correct one? Take 2 minutes, read the question carefully, then try to answer the question. After you try, then I will show you what is the correct answer, and then we will discuss about it. Let me check now the chat box if there are any answers for this particular question. Okay, I think I have few answers. Okay, I have number D, number D, number C, number D. So I have few number D and few number C. Okay, let us check and then let us discuss. If you have any doubt, any query, as I am saying again, if you have any doubt, any query during the session, if you have any problem hearing my voice or seeing the PPT, please write in the chat box. Okay, all this PPT and this presentation, this video, everything will be shared to you, so you don't have to worry about network connectivity from your side, and from my side I will try to be as fast as I can. So let us say what is the correct answer. The correct answer is number D, the pharmacophore. So now let us understand what each and every this term means. Okay, what is pharmacokinetics, what is pharmacodynamics, what is chromophore, what is pharmacophore. Let us understand. So first, pharmacodynamics. Pharmacodynamics is the study of the mechanism of action of drugs, and it explains. Okay, somebody wants to join. So pharmacodynamics is the study of the mechanism of action of drugs, and it explains what the drug does to the body and how they do it. It tries to explain the action effect relation as well as the dose relation effect. It involves the drug receptor interaction to elicit the action of the drug on the body. Pharmacodynamics is the study of the biochemical and physiological effect of the drugs and their mechanism of action. It provides the basis of the rational use of drugs and design for the better effective drugs. What is pharmacokinetics? Is the quantitative study of the drug movement in, throughout and out of the body. It is the study of the process by which a drug is absorbed, distributed, metabolized, eliminated by the body. The absorption, distribution, metabolism and excretion of a drug all involves its passage across the cell membranes. Pharmacokinetic properties are affected by the route of administration and the dose of the administered drug. Okay, so basically what is the difference between pharmacokinetics and pharmacodynamics, if I have to tell you in a very simple language. So pharmacokinetics is what it's a study about what our body is doing to a particular drug. You are taking a drug, and the drug, now what happens to that drug? Okay, what our body is doing to the particular drug, it is getting absorbed, then it is getting distributed, then it is getting metabolized, it is getting excreted. Okay, how this all these things, this is the pharmacokinetics, what our body is doing to a particular drug. Now what is the pharmacodynamics? The pharmacodynamics is what the particular drug is doing to our body, what is the effect. Okay, what is the action of the particular drug, what is the effect of the dose of the particular drug to our body, our system. So in short, pharmacokinetics is what a drug is doing to, what our body is doing to a drug, and pharmacodynamics is what the drug is doing to our body. Okay, so what is a chromophore? So basically it is a Greek word. Okay, the chroma means color and phore means bearer. So it is defined as any isolated covalently bonded group that shows a characteristic absorption or of electromagnetic radiation in the UV or visible region. Okay, a compound containing chromophore is known as chromogen, like C double bond C, C double bond O, nitro, these all are the chromophore. Okay, or chromophore contain, or chromophore is a part. So chromophore is the groups which is reason for a particular color of a particular compound. So now finally, what is a pharmacophore? So the IUPAC definition of pharmacophore is, it is an ensemble of steric and electronic features that is necessary to ensure the optimal supramolecular interactions with a specific biological target and to trigger or block the biological response. Okay, this is hydrogen, which is in drug design. The term pharmacophore refers to set of features that is common to a series of active molecules. Hydrogen bond donor and acceptor, positively charged, negatively charged groups and hydrophobic regions are the typical features. We will refer this is a pharmacophoric group, like hydrogen bonding, donor, acceptor, are all these things are taken in the pharmacophore. So very important, or very, we can say, there is a series of drugs, okay, or a series of molecule which are effective for a particular drug. So when we do this kind of SAR or structure activity relationship, no, so what we do is we keep one particular portion unchanged, okay, we just vary other groups, okay, as I have drawn in the last slide also, that let's say there is a phenol or there is a phenyl ring, okay, sorry, just let me erase and draw it better. So let's say there is a phenyl group, and let's say it is your lead compound, okay, this is a lead compound your, and this is a phenyl group, this is a R1, there is a R2, and there is a R3. So when we will do the SAR or structure activity relationship, we will keep this portion or this center unchanged, and then we change this R1, R2, R3, okay, and this particular through the reason of keeping this portion unchanged is this portion is first we have to identify if it is the particular, which is the particular portion, that is a other thing, but after identification of this particular, so we will keep this thing unchanged. So this is known as pharmacophore, okay, and this, after this orientation of this hydrogen bond, donor, acceptor, and every hydrophobic groups around this pharmacophore, it is a basic of this particular SAR, okay, and these groups are known as pharmacophoric group. So that's how we will learn slowly about this pharmacophore, this all these things I will show you slowly by this various examples, so you don't have to worry. So most of you have identified the answer correctly. So now we will move toward the next problem. So problem number 6, it says that the fibrous and globular proteins are associated with dash and dash structures of the protein respectively. Your options are number A secondary and primary, number B primary and tertiary, number C secondary and tertiary, and number D tertiary and secondary. You have to fill in the blanks, okay. What is the fibrous and globular proteins are associated with which structure of the protein in a respective manner. Is it secondary or primary, or is it primary tertiary, secondary tertiary, or tertiary secondary? You have 4 options A, B, C and D. Again all of you try to answer this question. The fibrous and globular proteins are associated with which structures of proteins? Secondary primary, primary tertiary, secondary tertiary, or tertiary secondary? All of you try to answer this question in the chat box. Even if your answer is wrong, it doesn't matter. I will give you 2 minutes. After that I will check and I will show you what is the correct answer and we will discuss. Okay, somebody wants to join. I think I have few answers for this question also. Number C, number C, number C, number C, number A. I have few number A, few number C and few number A. Okay. I will give you 2 more minutes and then we will discuss it. Okay, let's check what is the correct answer and then we have to discuss about the fibrous and globular protein also. So the correct answer is very good, number C, it is secondary and tertiary. So what is a fibrous protein? So when amino acids are joined in the straight chain and make a fibrous and sheet-like structure, then it is called the fibrous protein, like this. And when a polypeptide chain of amino acids became folded and makes a globular structure, ring and spherical, then it is called the globular protein. Okay, and these are associated with the fibrous structure is proteins are associated with the secondary structure of protein, and the globular protein is associated with the tertiary structure of protein. So we all know there are four structures of the protein, right? The primary, secondary, tertiary, and your quaternary structure. So what are the structure is all about? In the primary structure, what happens? There is a polypeptide chain, right? So how this polypeptide chain happen? We have amino acids. All of you, we know that the unit of the particular proteins are the amino acid, the basic units are the amino acids. So the amino acids, we have this amine group and as carboxylic acid group in a particular molecule, and one amine group of amino acid is associated or it linked with other carboxylic acid group of other amino acid unit, and they form a amide one, which is known as a peptide bond. Okay, so basically what happens, this is a very easy, or we all have read it previously. I am not discussing it in detail, but I just will show you like this. So let's say this is R1, this is, say this is R2, and we have this NH2, and we have this carboxylic acid. So this is, let's say it is a one amino acid, and we have similarly another amino acid where we have this carboxylic acid group here. So this amine and carboxylic acid group, we have this minus water, right? And we form this peptide linkage. So what is the structure of peptide linkage? We have this kind of carboxylate group, and then we have this NH group, and we have this is the particular peptide. So there are few features of this peptide linkage also. Let me discuss this all things again. So all this carboxylic acid, oxygen, and this will be 180 degree to each other. Okay, and this is the particular peptide bond. That's how so on it happens. So what happens during the secondary structure is, in the secondary structure, this particular polypeptide chains, which is the primary structures, they interact with each other. Okay, via hydrogen bond formation or any other methods, they interact with each other, and they form alpha helix and beta pleated sheets. Okay, and these are associated with this fibrous protein. And then after this fibrous proteins, or this alpha helix and beta pleated sheet, what we have this interact with each other via various interactions, and we have this tertiary structure of the protein, which is known as the globular protein also. Okay, and then we have this quaternary structure where the various tertiary structure interact with each other, and this is, and so on. So what is the differences between this fibrous protein and the globular protein? So the first, the fibrous protein, these are a class of insoluble proteins which constitute the main structural elements of the body. It has a long and narrow shape and have a helical or sheet-like structure. Whereas the globular protein, a protein soluble in water and tend to be involved in metabolic functions. It has a round or spherical shape and have a folded like ball-like structure. Okay, the fibrous protein has a secondary structure. It is the secondary structure of the protein, and the globular in the tertiary structure. The fibrous protein consists the repetitive amino acid sequence, but in the globular protein we have irregular amino acid sequence. In the fibrous protein, it is insoluble in water, acid, and bases, and have a strong intermolecular interaction, whereas in the globular protein, they are soluble in water, acid, and bases, and have weaker intermolecular interaction. Fibrous proteins are less sensitive to the changes of temperature and pH and involved in forming structures like collagen, elastin, actin, myosin, fibrin, keratin. Okay, and the very good example of the particular fibrous proteins are the keratin, which is present in the hair, we all know, and the collagen, elastin, which are present in the skin. So all of these are made of the fibrous protein. Whereas the globular protein, these are sensitive to the changes of temperature and pH and involved in mainly in metabolic reactions like enzymes, hormones, hemoglobin, immunoglobulin. All of these are the example of the globular protein. Okay, so this is a very important, this differences and similarities or difference between this fibrous and globular protein. Okay, so try to remember these things. So now I will move toward the next problem. So this problem says that which of the following statement is false? for the determination of the 3D structure of the proteins. Number A, best way is to crystallize the protein. Number B, NMR can also provide the static conformation of proteins. Number C, cryo-electron microscope can directly show the image, no need for crystallization. Number D, for NMR, highly concentrated solutions are needed. So, which of the following statements is false for the determination of 3D structure of proteins? Read the options carefully, read the question carefully, then try to answer it in the chat box. Generally, in case of this kind of true false kind of question or this kind of correct incorrect kind of question, many students made mistakes, including myself. I have also made many mistakes while reading this kind of questions. So, during this this type of questions, please pay attention what is asking for. It is asking for the true, for false or correct or incorrect. So, try to understand and then try to answer. So, what is the which of the statements, which of the following statements is false for determination of the 3D structure of proteins? Number A, the best way to crystallize a protein, it is the best way to crystallize a protein. NMR can provide static conformation of protein. Number C is cryo-electron microscope can directly show the image, no need for crystallization. Number D, for NMR, highly concentrated solutions are required. Let me know in the chat box which of the following options is false for the determination of 3D structure of protein. Take few minutes, then I will discuss the answer. Let me check the chat box if I have any answer till now. Okay, I have few answers. Number C, number D, number D, number D, number D. Okay, most of the answers are related to number D. So, let me check if number D is correct or not. So, the correct answer means the false statement regarding the determination of the 3D structure of proteins is number B. Okay, NMR can also provide the static conformation of proteins. It is not correct. It is the false statement. Now, let us discuss. The 3D structures are precious source for the informations. So, whenever we are studying the particular, as I have the target validation and the hit identification, I have mentioned that the protein modeling is a very important method. Okay, and so for this protein modeling, what we need? We need the 3D structure of the protein or the information for the 3D structure of the protein. You will see there are few there is a very important website or very important database. I can say not website, important database is the RSC PDB or protein data bank. Many of you may have heard about this protein data bank where the 3D structures of the proteins are deposited. Okay, if you are particularly targeting a disease, you have to check in that PDB or protein data bank if the 3D structure is available, because these are the very important sources of information for a particular target. Okay, so what happens? Why? Because from the 3D structure we can determine the shape and domain structure, the protein classification, prediction of the function of uncharacterized protein, interaction with other micromolecules, interaction with small ligands, metal ions, nucleotides, substrate, cofactors and inhibitors, evidence for enzyme metabolism, structure based drug development, the post-translational modification disulfide bonds like in glycosylation, experimental evidence for transmembrane domain. So these are the important sources of information which can be we have from the 3D structure of protein. So regarding determination of the 3D structure of protein, there are different techniques. To determine the 3D structure, one best way is the X-ray crystallography. So if you can isolate a crystal structure of a protein, then it is the ultimate structural evidence. Methods of determination, the complete 3D structure. Usually there are three types. One is X-ray crystallography, that is the most reliable one. Then we have the NMR spectroscopy. Very high field NMR machine is needed for this, and through NMR spectroscopy you can determine the 3D structure of protein. However, there are some disadvantages of NMR spectroscopy. You cannot handle very large molecules in NMR, but at the same time there are some advantages as well. In X-ray crystallography you cannot crystallize a protein, then you cannot get the crystal structure. X-ray crystallography needs crystal and the structure is a static one. So we can have this average structure in the crystal, but in NMR it is recorded in the solution. So when you see exactly the dynamics of the system, okay, but you need a highly concentrated solution to get the signals. The peaks are usually very complicated. You should isotopically label carbon, isotopically label nitrogen, and this technique to do that. But NMR you can do the structural analysis, but not for a very large protein. At up to 60 kilodalton you can do the structural elucidation. So NMR does not provide the static conformation. Why? Because NMR we take in a solution. Okay, for NMR you have to dissolve that particular compound or particular protein in a particular solvent. Okay, and this particular solvent or the solution, this is in a dynamics formation. Okay, the proteins or these things are not static like in the crystal or X-ray crystallography. The compound, the protein, everything is in the static position. Okay, it does not move in the crystals, but in solution it moves. So the NMR provides us the dynamic conformation of the proteins, not the static one. But as it is said that the NMR is recorded in solution, but you have to have very concentrated, otherwise the peaks won't come very properly. Okay, and you have very complicated NMR for the proteins, and to determine the NMR you have isotopically labeled atoms like carbon, hydrogen, nitrogen, all you have to use isotopically labeled, otherwise you won't be able to solve the particular NMR of a protein. Okay. So I think it is clear. If you have any doubt for this question and any other question, you can still let me know in the chat box. I will repeat whatever doubts you are having for any question, just write it in the chat box. At the end of the session I will repeat all those things. Okay. So now I will move to the next question. Problem number 8 says, which of the following is not a weak interaction? Your options are: number A, pi-pi stacking, number B, hydrogen bonding interaction, number C, salt bridge interaction, and number D, disulfide bond. So which of the following is not a weak interaction? Pi-pi stacking, hydrogen bonding interaction, salt bridge interaction, disulfide bond. Which of the following is not a weak interaction? Somebody wants to join. Okay, I think you have just started to give the answers too, which is good. Okay, take 2 minutes, then I will discuss. Which of the following is not a weak interaction? Pi-pi stacking, hydrogen bonding interaction, salt bridge interaction, disulfide bonds. I will check after a minute and then we will discuss it. Which of the following is not a weak interaction? Let me check the chat box, then we can discuss it. Okay, so I have multiple answers I think for this question. Let me check. Okay, number C, number A, number D, number C, number D and number A are the most given answers. Let me check and see which is the particular answer. So, the correct answer is number D, disulfide bond. Disulfide bond is not a weak interaction. Why? Now, let's say this is a pi-pi stacking interaction. Okay, the pi electron cloud of a particular ring or a particular aromatic ring interacts with the pi electron cloud. When it interacts with the pi electron cloud of the particular other aromatic ring, it is known as the pi-pi stacking. Okay, then this is the hydrogen bonding interaction. So, hydrogen bonding interaction and salt bridge interactions are basically electrostatic interactions. Okay, so this is the salt bridge interaction. So, what is the salt bridge interaction? So, we have this amine, all we know this particular amino acid, this stay as a zwitter ionic form, right, and we have acidic and basic amino acids also, right. So, this is the particular, when there is a acidic and basic amino acids, these amino acids are charged, like basic amino acids are positively charged and acidic amino acids are negatively charged. So, the interaction between these positive negative charges are known as the salt bridge interaction. Okay, and the hydrogen bonding interaction is when a particular hydrogen bond donor is interacting with a hydrogen bond acceptor. So, this is a hydrogen bonding interaction. All these things are interactions, right. All these things are either these are electron crowding interaction or these things are any kind of electrostatic interactions. Okay, there is no particular bond formation. Okay, but in case of disulfide bond, we have this particular covalent bond formation. Okay, so how this disulfide bonds are formed? So, we know there are sulfur containing amino acids like cysteine. Okay, so what happens, this two cysteine residues, they can form this kind of SS sulfur sulfur covalent bond. Okay, or this is known as this disulfide bond. So, what is the reaction? Two cysteine residues, they react and the two electrons are emitted and two hydrogens are also, protons are also emitted in this particular reaction. This bond is known as disulfide bond and it is a proper covalent bond which has a very strong interaction. Okay, so this is not a weak interaction. So, remember, disulfide bonds are very strong interaction, these are not the weak interactions. Okay, unlike pi-pi stacking, hydrogen bond salt bridge interactions, which are the interaction, no covalent bond formation happens during pi-pi stacking, hydrogen bonding or salt bridge interaction. Okay, so this was the problem number 8. Now, moving forward, problem number 9 says identify the correct statement. Number A, in the interior of globular protein, hydrophilic amino acids are present. Number B, hydrophobic amino acids stay in the interior of the globular protein. Number C, in the exterior both hydrophobic hydrophilic amino acids reside. Number D, there is no concrete rule regarding this. So, read all the options carefully and then try to answer it in the chat box. What is the correct statement? Number A, in the interior of globular proteins, hydrophilic amino acids are present. Number B, hydrophobic amino acids stay in the interior of globular protein. Number C, in the exterior of this globular protein, both hydrophobic hydrophilic amino acids reside. And number D, there is no concrete rule regarding this. So, read the options carefully, identify which of the following is the correct one and then let me know in the chat box. A, B, C or D, which of the following you think is the correct option for this problem number 9. Okay, so I have few answers. number B, number C, number D, number A. Okay, so I think for this question I have A, B, C, D, I mean 4 as answers. So which is good that we have this kind of discrepancy. Okay, let me show you the answer and then let us discuss. Okay, so the correct statement is hydrophobic, number B, hydrophobic amino acid stays in the interior of the globular protein. Okay, this is the correct statement. Now let us discuss. First we have to know the classification of the amino acids based on the polarity. So first we have this non-polar amino acids, which are the hydrophobic amino acids like glycine, alanine, valine, cysteine, proline, leucine, isoleucine, methionine, tryptophan, phenylalanine. So these are the non-polar or hydrophobic amino acids. And one more thing, whenever you are reading about the amino acids, try to memorize this 3 letter codes and also the single letter code for this amino acid, like glycine is GLY and G and all and so on, because there are many questions or many times you have to only there will be this one letter code also, you have to identify which is the particular amino acid from that code. So try to remember that as well. Okay, so these 10 are the hydrophobic or the non-polar amino acids. Then we have polar amino acids, which are considered as hydrophilic. Okay, so polar amino acids can be of 3 types. One is the uncharged, and then we have positively charged and then negatively charged. So what are the, for the uncharged we have this serine, threonine, tyrosine, asparagine and this glutamine. So why these things these are uncharged and these have this particular polarity? Because of this for this hydroxyl group in serine, threonine and tyrosine. Okay, and this amide group in asparagine and glutamine. That's why this is called hydrophilic. For positively charged we have 3, lysine, arginine or histidine, which are also known as the basic amino acids. Okay, so basic amino acids have this positively charged side chain. And then we have this 2 negatively charged amino acids, which are the aspartic acid and glutamic acids, which are the acidic amino acids. Okay, so this is the total classification of the amino acids based on the polarity. So now what is the globular protein? So we have discussed the globular and fibrous protein before also. So globular proteins are named for their approximately spherical shapes and are most abundant proteins in nature. These globular proteins exist in enormous variety of the 3-dimensional structures. Nearly all globular proteins contain substantial numbers of alpha helixes and beta sheets folded into a compact structure that is stabilized by both polar and non-polar interaction. The globular 3-dimensional structure forms spontaneously and is maintained as a result of interaction among the side chains of amino acids. Most often, the hydrophobic amino acid side chains are buried closely packed in the interior of globular protein out of contact with water. Hydrophilic amino acid side chains lie on the surface of the globular proteins exposed in the water. Consequently, globular proteins are usually very soluble in the aqueous solutions. Okay. Now, the diversity of the protein structures reflect the remarkable variety of functions performed by the globular proteins like binding, catalysis, regulation, transport and immunity, cellular signaling and more. So why we are reading about this globular proteins very well? Because this most of the enzymes or drug targets are made for of this globular proteins. Okay, so remember this globular protein has 3-dimensional structure form, and which is amino for this mineral is the hydrophobic amino acids. I will just highlight this portion for your reference. Okay, so remember that the hydrophobic amino acid side chains are buried closely packed in the interior of globular protein out of the contact of water. Hydrophilic amino acids are side chains right on the surface of globular proteins exposed in the water, and that's why these globular proteins are soluble in water, since hydrophilic amino acids are in the exterior of this particular globular protein. Okay. Somebody wants to join. So we have 4 different answers for this question, but which is fine. So I think it is clear to all now what is the globular protein and how the amino acid reside, and what is the classification of amino acids based on the polarity. So now I will move toward the next problem. Problem number 10 says which one of the following statement regarding the enzyme is not true. Number A, enzymes are one kind of proteins. Number B, they can change the rate of the reaction. Number C, they get consumed in the course of the reaction. And number D, they are site specific in nature. So, which of the following statement regarding the enzyme is not true? I think it is a very easy question and I want all of you to answer this question in the chat box. I will just give you a minute because I know all of you can answer this question very correctly. So, just write in the chat box A, B, C or D, which of the following is the correct answer, means which of the following is not true regarding enzyme. Number A, enzymes are one kind of protein. Number B, they can change the rate of reaction. Number C, they get consumed in the course of reaction. Number D, they are site specific in nature. Let me check the chat box now. And I know all of you have answered it correctly. Okay. So, now I will show you and let us discuss about a little bit about enzymes. So, the correct answer is yes, number C, it is not true that they get consumed in the course of reaction. So, how does the enzyme work? So, this is an enzyme and this is a substrate, and in the enzyme it is a particular protein which change the rate of the reaction and they are site specific. So, in the enzyme we have a particular site which is known as the active site, and the substrate which can bind in the active site of this particular enzyme can only catalyze the particular reaction for those substrate. Okay. So, first there is an enzyme, there is active site and substrate, and the enzyme substrate complex is formed. After enzyme and substrate complex has formed, so then what will happen, there will be the reaction, particular reaction, and then we will have this particular product, but in the course of the reaction this particular enzyme won't get consumed. Okay, they will be intact at the end of the reaction. So, particularly the protein, it is a protein with catalytic properties due to the power of specific activation, biological system sensitive to temperature change. In case of rate of reaction without increasing the temperature, in case increase the rate by lowering the activation energy, they create a new reaction pathway. They are globular proteins with complex 3D structure. The active site is where the reaction is catalyzed. So, protein, basically these particular enzymes, how they alter the rate of the reaction by lowering the activation energy. Also actually particularly temperature, by increasing the temperature also we can catalyze the particular reaction, but since the biological systems are sensitive to temperature change, these enzymes do not alter the temperature of the reaction. Okay, but they only lower the activation energy, thus increasing the rate of the reaction. And as I have mentioned in the last slide only, all the enzymes, they are the globular protein with complex 3D structure. Okay, and they are site specific because in the particular there is particular conformation of active site, and the particular substrates only can bind to that particular active site of an enzyme. Okay. Now, problem number 11. It says which of the following statement is false regarding trimotrypsin. Number A, it preferentially cleaves the protein chain at the carboxyl site of amino acid. Number B, the mechanism is which it works is the acid-base catalysis. Number C, the mechanism by which it works is nucleophilic substitution. And number D, catalytic triad of serine, histidine and aspartate is present in the active site of the trimotrypsin. So, identify which of the following statement is false regarding trimotrypsin. Basically, trimotrypsin is a particular enzyme, okay, which cleaves the proteins, but where it cleaves I won't mention right now. So there are 4 options regarding this chymotrypsin. Number A, it preferentially cleaves the protein chain at the carboxyl side of amino acid. Number B, the mechanism by which it works is the acid base catalysis. Number C, the mechanism by which it works is the nucleophilic substitution. And number D, catalytic triad of serine, histidine, aspartate are present in the active site. Now identify which of the following is the false regarding chymotrypsin. I will give you 2 minutes and then I will show you the answer and we will discuss. Which of the following statements is false regarding chymotrypsin? Let me check the chat box now. If I have any answer, then we will discuss it. Okay, so I have few answers, number C, number B, number C, number C. So I think most of you have said it is number C and one I have I think number B. Okay, let me check which is the correct answer. So the statement which is false regarding the chymotrypsin is number C. The mechanism by which it works is the nucleophilic substitution. Now let us check. So basically chymotrypsin is a particular enzyme which preferentially cleaves the protein chain at the carboxylic site of aromatic amino acids, okay, and there is a catalytic triad of 3 amino acids in the active site. Which are the amino acids? We have a aspartate unit, this is the aspartate unit, this is the histidine ring of a particular it's the imidazole ring of the particular histidine, and then we have the serine, the phenolic OH of the serine, okay. And how this particular catalytic triad happens, this aspartate unit is interacting with this histidine unit and then this histidine unit is also interacting with the serine unit. So we have this kind of hydrogen bonding interaction of these 3 amino acids, okay. So how this particular catalysis happens? So first we have this oxyanion hole, we have this oxyanion hole. So this is a particular amino acids and this serine unit first this acidic aspartate group, it pulls this proton of this serine group toward itself, okay, and this histidine helps in doing it, and that is how this kind of intermediate forms where this serine proton is interacting with this histidine ring, and then it this O becomes negative and it attacks this particular carboxylic acid carbon, okay, and this tetrahedral intermediate is forming. Then we have this acyl enzyme and this acyl enzyme is formed, which is the particular acyl group of this amino acid interacts with this serine, okay, binds with the serine. Again we have this particular histidine which attack, there is a water molecule which attacks on this particular acyl enzyme, and again one tetrahedral intermediate is formed and we have this cleaved carboxylic acid part and we have this enzyme as it is again, okay. So first it starts from here, this is the first step where this particular amino acid side chain comes into the active site, and then this catalytic acid base catalysis happens here. This aspartate group, histidine group and serine, it goes through the acid base catalysis, and then this nucleophilic attack happens on this particular peptide chain, and this cleavage happen and regeneration of this particular enzyme happens after the addition of water. So that is how this particular catalytic cycle happens. So this mechanism does not work by nucleophilic substitution. Yes, you may say that there is a nucleophilic attack and tetrahedral intermediate formation, okay, but the basics of this particular attack is the acid base catalysis. Without this acid base catalysis or acid basic assistance of this aspartate and histidine, this OH of the serine won't be able to attack this peptide linkage, okay, or won't be able to cleave this peptide linkage. So this is a mechanism works by acid base catalysis. So now I will go to the next question. Problem number 12 says the correct graph for an enzyme controlled reaction versus substrate concentration is number A, B, C and D. So I think this is also very easy for you, whoever have learned a little bit about the enzyme controlled reaction or the catalysis kinetics, enzyme kinetics, they will be able to answer this question. So I will give you 1 minute. In this 1 minute I want all of you to answer this question. The correct graph for enzyme controlled reaction rate and the substrate concentration. So identify the correct graph for the enzyme control reaction rate and substrate concentration. Let me check the chat box. I think many of you have answered it by now. Okay, so I have different answer. I think I have let me check. So I think I have few number C, few number A, number C, number A. So number C and number A are the most given answers. Let me check which of the following is correct. So the correct graph for the enzyme control reaction rate versus substrate concentration is number A. This particular graph, it is the graph for the enzyme control reaction rate versus the substrate concentration. So in the x axis we have the substrate concentration, okay, and on the y axis we have the velocity of reaction or the rate of the reaction, and this is known as very famous equation, rate equation, or very famous kinetics we know for this enzyme control reaction. Can anybody tell in the chat box what is this particular enzyme kinetics is called, or this graph is called? I think many of you know. There is a very famous equation and very famous particular for this graph and kinetics. Yes, Michaelis-Menten equation. This graph is for the Michaelis-Menten equation for the enzyme kinetics. Very good. We have a few more enzyme kinetics graphs present, which we will discuss in the next class. Okay. So this graph is known as Michaelis-Menten enzyme kinetics graph, okay, and this is the particular curve as you can see. So I will discuss it, okay. So first portion, it is the, here there is a lots of enzymes are available in the first portion. So initially there is substrate concentration is low, okay, but enzyme concentration is constant, okay, and the substrate concentration is low. So what happens, all the in substrate is saturated with the enzyme, and the first order kinetics is followed in this first portion. Then we have some mixed order kinetics, where there is a competition between the enzyme molecules, because there are, the substrate concentration is increasing but enzyme concentration is almost the same, okay. And then we have this zero order kinetics. Why zero order kinetics? Because we have few enzymes, all enzymes are occupied with the substrate concentration. So now increasing the substrate concentration now won't increase the particular rate of the reaction. So the order is the zero order. So first we have this first order, then we have the mixed order, and then we have the zero order. Why first we have the first order? Because increasing the substrate concentration increases the amount of the enzyme substrate complex, okay, and when all the enzymes are occupied or all the enzymes are form the enzyme substrate complex, by increasing the substrate concentration does not alter the particular rate of the reaction. So it is the zero order, okay, and this is called known as the Vmax. If you just pull a line or draw a perpendicular on y axis from this plateau, so we will have this particular Vmax, which is known as the maximum rate of the reaction, or Vmax. Okay, and this is known as the, and if you just draw a particular from this half point, this Vmax by 2, from this half point, if you draw a particular perpendicular on the curve and then join it on the x axis, then we have a particular substrate concentration, okay, which is known as Km. And what is the Km? Km is the Michaelis-Menten constant. All of this, all this equation, all this mathematics we will gradually discuss, so you don't have to worry about it. So this is, just remember, this is known as the enzyme control, how this graph looks like, the enzyme control reaction versus substrate concentration. Okay. So now moving forward, problem number 13 says, in ADME, A stands for number A, agonism, number B, antagonism, number C, absorption, and number D, affinity. ADME I have discussed at the previously when I was discussing about the pharmacokinetics, I think. So I have coined this term ADME also, and I think I have discussed what A stands for. Now let's see how many of you remember and how many of you know what is A stands for in ADME. Agonism, antagonism, absorption, affinity. Let me know in the chat box. In ADME, A stands for which? Let me check the chat box. Yeah, ADME in pharmacokinetics. I have mentioned ADME in pharmacokinetics. Number C, number C, number C. So all of you have told me that it is number C. Let me check if it is correct or not. So very good, it is number C. Absorption stands for in ADME. So what is absorption? Absorption is the transfer of a drug from its site of administration to the bloodstream. Most of the drugs are absorbed by the way of the passive transport. Intravenous administration has no absorption. Fraction of administered drug and the rate of the absorption are important in case of pharmacokinetics parameters. Okay, so absorption is one of the first parameter for pharmacodynamics. So let's say for in case of drugs which are taken orally, okay, so first the drug is absorbed in the body, okay, till the drug is go to the from the point of administration, it goes to the main bloodstream. So in case of intravenous administration, means the drugs which are given through the injection, there is no absorption because it is directly injected into the bloodstream. This is the overall pharmacokinetics chart we can say. So first we have this absorption, okay, oral drug is administered, okay, and the drug is released in the GI tract, which is the gastrointestinal tract, and resistant to gastric acidity prodrug for better absorption. Okay, 1 second. Okay, so the first, when the drug is oral drug is given, sometimes prodrugs are given. Why prodrugs are given? Because sometimes the drugs are they have this resistance to gastric acidity, so for better absorption prodrugs are given, okay, and sometimes extended release formulations are given to sustained and delayed release. So first the drug is given, it is absorbed in GI tract by the diffusion and active transport, okay, then it is distributed in the body. So the distribution can happen in various ways, okay. I will discuss all this pharmacokinetics later in detail. So we have this kind of deep compartments like bone, adipose tissues, okay, and then accumulation can happen in the CNS. The drugs are so the drugs which are given, they are from central nervous system, by going to central nervous system, it is blocked by the BBB or blood brain barrier, okay. Now, lipophilic drugs, they can cross the BBB and they are poorly dialyzable, okay. And then we have this fast pass efficacy bioavailability is low. Then we have this metabolism. Metabolism can happen by two methods, phase 1 metabolism, phase 2 metabolism, and all this metabolism, after this we have this kind of excretion. Okay, after distribution we have this metabolism and we have this excretion, and now this excretion can have we can have this excretion by kidney. Okay, and like hydrophilic drugs, high molecular mass molecule, fast effect, and these are this can be the kidneys can be given excretion. And then after the phase 1 and phase 2 metabolism, sometimes happen there are phase 2 metabolite which goes to the phase 1 metabolite and again it is reabsorbed. So all this phase 1 metabolism, phase 2 metabolism I will discuss in detail. So just remember there are 4 important terms at me, like absorption, distribution, metabolism and excretion. In absorption, the drug is absorbed in the GI tract. For oral administration, it has a diffusion and active and passive transports. Okay. Then moving toward the next problem. Problem number 14 says, what is the reason of the lower pKa value of phenol than cyclohexanol? Option A, cyclohexanol is more thermally stable. Option B, pi electron cloud of aromatic ring takes part in the delocalization. Number C, the specific reason cannot be determined experimentally. And number D, cyclohexanol has more chances of hydrogen bond formation. What is the reason of lower pKa value of phenol than cyclohexanol? Let me know in the chat box which of the following you think is the correct answer. What is the correct reason for the lower pKa value of phenol than cyclohexanol? Number A, cyclohexanol is more thermally stable. Number B, pi electron cloud of aromatic ring takes part in delocalization. Number C, specific reason cannot be determined experimentally. And number D, cyclohexanol has more chances of hydrogen bond formation. A, B, C or D, which of the following you think is the correct answer? Let me check the chat box. Number B, number B, number A. So most of the answers are given like number B and number A. Okay, let me check and let us discuss. So the correct reason of lower pKa value of phenol than cyclohexanol is number B, pi electron cloud of aromatic ring takes part in delocalization. So lower pKa value means phenol is more acidic than cyclohexanol. Okay. So this is the structure of cyclohexanol, the pKa value is 16. This is the structure of phenol, the pKa value is 10. Lower the pKa means lower the acidity of this proton. So this is the localized anion of the cyclohexanol, and this is the phenoxide ion, okay, for the in case of phenol. So even the delocalization into hydrocarbon part of a molecule increases the acid strength. In phenol, the phenolic OH, in phenol the OH group is directly attached to the benzene ring. On deprotonation, the negative charge can again be delocalized not onto the other oxygen atom but onto the aromatic ring itself. The effect of this stabilizes the phenoxide anion relative to the conjugate base of the cyclohexanol, while no delocalization is possible, and this is reflected in the pK values of two compounds, which is 10 for phenol and 16 for cyclohexanol. So since this anion or phenoxide ion is delocalized onto the on the particular pi orbitals or the pi system of the aromatic ring of the phenol, that's why this phenoxide ion has more stabilized. Since the this stability of conjugate base is increasing, the acidity of phenol is increased and the pK value is lower. Okay. So now problem number 15, it says that identify the incorrect statement. Number A, DMF shows two NMR signals for the methyl proton at room temperature. Number B, DMF shows the single NMR signal for the methyl proton at room temperature. Number C, DMF shows single NMR signal for methyl proton at high temperature. And number D, the NMR of the DMF shows time average signal at high temperature. So which of the following statement is incorrect? DMF means dimethyl formamide. Identify the incorrect statement. Number A, DMF shows 2 NMR signals for the methyl proton at room temperature. Number B, DMF shows single NMR signals for methyl proton at room temperature. Number C, DMF shows single NMR signals for methyl proton at high temperature. Number D, the NMR of DMF shows time average signals at high temperature. Let me check the chat box. Okay, I think I have few answers. Number B, number D, number A, number B. Okay, I have different answers. So let me show you and then let us discuss about this. This is a very interesting question, very important one too. So the correct answer, or rather the incorrect statement, is DMF shows single NMR signal for methyl proton at room temperature. This is the structure of DMF. DMF is the NN dimethyl formamide. So as you can see, the structure of DMF is like this. The NMR spectrum, so this is known as the influence of restricted rotation, okay, on NMR signals. So first let me show you. So this is the this is the NMR of the DMF. Methyl, so for this methyl, it is not zoomed one, but I think you can identify there are two peaks. There one smaller one and one is larger one in the methyl region. And this is for this, so for as this is for this aldehydic proton, for this proton, okay. This is the at the 8 is the peak, and from the 3 the peak is, there are two peaks, it is the room temperature. So why we have this two peaks for this methyl proton? Because we all know for the NMR these two are equivalent, right? But but still we have two single NMR peaks for methyl proton at the room temperature for the DMF. So why? Because the NMR of spectrum for DMF or NN dimethyl formamide recorded around room temperature, it shows two signal for methyl proton, although it might have been expected that the two methyl groups would be magnetically equivalent environments. Dimethyl formamide is represented by two resonance forms shown below. This is the two resonance form of DMF. It is the result of the conjugation between the carbonyl group and the nitrogen non-bonding pair is to increase the double bond character of the CN group sufficiently to restrict the rotation of at the room temperature. One methyl group, so since there is this is a lone pair, it is takes part in the resonance. So now this bond has a double bond character. Since this bond has a double bond character, now this rotation around this bond is not possible. So now we have two different methyl groups. One is cis to the oxygen and one is trans to the oxygen. The anisotropy of the carbonyl group is sufficient to influence the chemical shift of the position of the cis group. Using a heated probe of NMR instrument, the spectrum of NN dimethyl can be recorded at high temperature also. And when the temperature is high, it shows only one signal for the N methyl groups at the elevated temperature, because when there is a high temperature, the rotation around the CN bond is so rapid that each methyl group experiences the same time averaged environment. Okay, so at the high temperature it shows only one signal for the methyl proton, but in case of room temperature it shows two different peak for the methyl signal, and at the high temperature it shows a time average image for the time average image for the NMR spectrum. Okay, so that's why the number B was the incorrect statement. So now moving forward, problem number 16 says identify the incorrect statement among the following. Number A, coenzymes are either small organic molecule or inorganic metal ions. Number B, apoenzymes are activated by cofactors. Number C, prosthetic groups are coenzymes which bind permanently with enzymes. Number D, cofactor is non-protein molecule. So which of the following statement is incorrect? Number A, coenzymes are either small organic molecules or inorganic metal ions. Number B, apoenzymes are activated by cofactors. Number C, prosthetic groups are coenzymes which binds permanently with enzymes. And number D, cofactor is non-protein molecule. Identify the incorrect statement among the following. Let me check the chat box. Okay. So number C, number B, number C. So most of you think the number C is the incorrect one. Okay, let us check. So the incorrect statement is number A. Okay, coenzymes are either small organic molecule or inorganic metal ions. It is the incorrect statement. Let us understand what are coenzymes and what are cofactors. So a large number of enzymes require an additional non-protein component to carry out the catalytic functions, and these known as the cofactors. A coenzyme or metal ion is covalently bound to the enzyme protein is known as the prosthetic group. So cofactors are two types. First is the inorganic ions like the iron, magnesium, manganese, zinc, or a complex organic molecule which is known as the coenzyme. Okay, so coenzymes are the complex organic molecules, okay, not inorganic metal ion. Inorganic metal ions are not known as the coenzyme. Coenzymes are complex organic metal ions. Okay, some enzymes require both coenzyme and one or more metal ions for the activity. Coenzymes function as transient carriers of specific functional groups. So what are the types of the cofactors? So two cofactors can be essential ions or coenzymes. Coenzymes can be co-substrate which are loosely bound or prosthetic group which are tightly bound. Okay, essential ions can be activator ions which are loosely bound, and metal ions, so metalloenzymes which are tightly bound. So, okay, so coenzymes are the organic portion or the complex organic molecules. Metal ions are not coenzymes. Okay, apoenzymes are otherwise apoenzymes are activated by cofactor. It is the correct one. Prosthetic groups are the coenzymes which are permanently bind with the enzyme or which are the tightly bind with the enzyme, which is also correct. Cofactor is non-protein molecule. Okay, so cofactor, it is a non-protein molecule. Mainly the ions are known as the cofactor. Okay, these are not protein molecules, these are non-protein molecules. Iron or organic molecule, these are not protein molecule. Okay, so this was the, so number A was the incorrect one. Okay, coenzymes are not inorganic metal ions. So number 17, it says which of the following statement is false. Number A, both receptors and enzymes have the active site. Number B, only enzymes have active site, not receptors. Number C, after binding, reaction takes place in enzymes. Number D, after binding, no reaction takes place in receptors. So which of the following statement is false? Number A, both receptors and enzymes have active site. Number B, only enzymes have active site, not receptors. Number C, after binding, reaction takes place in enzymes. Number D, after binding, no reaction takes place in receptors. So identify which of the following is the false answer. Since we don't have much time left today, I will just do it quickly. So is there any answers for this question? Okay, I have many answers, I think. Okay, number C, number B, number C, number A. So I think A, B and C. I have 3 different answers for this question. So let me check. So the correct answer means the statement which is false, it is number B. Only enzymes have active site, not receptor. It is false. Okay. So what is a receptor? A receptor is a protein molecule usually found embedded within the plasma membrane surface of a cell that receives chemical signals from outside of the cell, and when such chemical signals bind to a receptor, they cause some form of cellular and tissue response. Difference between enzymes and receptors: in enzyme there will be an area which has a correct shape to accept the incoming messenger. The area is known as the binding site and it is analogous to the active site of the enzyme. Okay. The chemical messenger does not undergo chemical reaction. It fits into the binding site of the receptor protein, passes its message and leaves. So, the enzymes and receptor both have active site. Receptors also have a active site, okay, and this area is similar to the particular active site of the enzyme. But what is the difference? That the reaction takes place in the particular active site of reaction enzymes. But in case of receptors, there no particular reaction happens, okay, only messenger molecules comes bind to the active site of the receptor and it passes its message and leaves. There is no chemical reaction. Some receptor characteristics are: binding of ligand is only temporary, ligand binding produces physical changes in a protein configuration, initiating intracellular changes that ultimately generates behavioral effect. Receptors have a life cycle as other proteins do. Receptors can be modified in numbers, long-term regulation and in sensitivity. Receptors can upregulate, increase the numbers of chronic absence of agonist, or downregulate, decrease in number, which is chronic presence of a agonist. Okay. So, these are the difference between the receptors and enzymes and some characteristics of the receptors. Now, problem number 18, it says that which of the following statements is true? Number A, ion channels are called metabotropic receptors. Number B, neurotransmitters are involved in G protein receptors. Number C, G protein receptors do not couple with ion channels. And number D, all of the above. Which of the statement is true? Take 1 minute and let me know which of the following is you think is the correct one. Number A, ion channels are called metabotropic receptors. Number B, neurotransmitters are involved in G protein receptors. Number C, G protein receptors do not couple with ion channels. And number D, all of the above. Let me check the chat box. Okay, number D. Most of the answers are related to number D, I think. Okay, let me check if number D is the correct one. So, the statement which is true is number C, G protein receptors do not couple with ion channel. It is the only true statement. Okay. So, what are the G proteins? G proteins are known as the guanine nucleotide binding proteins involved in transmitting signals and function as molecular switch. G protein coupled receptors like GPCRs are also known as the 7 transmembrane receptor or 7T receptors or serpentine receptor or G protein linked receptor or GPLR. It constitutes a large protein family of receptors that sense molecules outside of the cell and activate the inside signal transduction pathways and ultimately cellular processes. So, what are the differences between ligand gated ion channels versus G protein coupled receptors? So, a group of transmembrane ion channel proteins which open is to allow such as sodium, potassium, calcium or chloride to pass through the membrane in response to binding to chemical messenger such as neurotransmitter is known as ligand gated ion channels, whereas the proteins located in the cell membrane that binds to extracellular substances and transmit signal from this substance are known as to an intracellular molecule is called a G protein. So, these ligand gators are called the ionotropic receptors and G protein coupled receptors are called the metabotropic receptors. So, the first statement is false. Okay, ion channels are called ionotropic receptors and G protein coupled receptors are called the metabotropic receptors. The multimers are made of at least of 5 or 4 individual protein subunit. The 7' transmembrane domain receptors is present in G protein coupled receptor. These couple with ion channels, okay, and they do not couple with ion channels, involve in direct opening of the ion channels after binding to a neurotransmitter. But G protein coupled these are involving indirect binding of ion channels with metabotropic activation of G protein. So neurotransmitters are not involved in G protein receptors, they are involved in ion ligand gated ion channels, okay. So only statement number C is the correct one or the true statement. So now problem number 19, it says that ankymeric assistant is commonly known as number A, neighboring group elimination, number B, electrophilic substitution, number C, addition and elimination, and number D, neighboring group participation. I think many of you have heard this term ankymeric assistance. Now try to answer this question. What is the more common term for ankymeric assistance? Number A, neighboring group elimination, number B, electrophilic substitution, number C, addition elimination, number D, neighboring group participation. I will give you a minute, then I will show you the answer and we will discuss it. So let me check, because I think this is a very common question. Okay, I have different answer. I think most of the answers are like number D, okay, and then we have number B, number C also. Okay, I have different answers for this question. Okay, let us check. So ankymeric assistant is commonly known as neighboring group participation, which is the option number D. Neighboring group participation is occasionally known as ankymeric assistant. It is a Greek word where the anky means neighboring and mar means part. So as you can see these kind of examples, okay, this molecule reacts with water 600 times faster than this. This again reacts with water, reacts with CF3COOH 3000 times faster than this, and so on. So why this particular, what is the difference between these and this? This sulfur, right? What is the difference between this and this? This phenol, this phenyl green. What is the difference between these two is the double bond here, and what is the difference between these two is the particular substitution or the pattern of this cis trans of this particular groups. So why this happens? This neighboring groups actually evidently increase the rate of the substitution reaction significantly. So this is the example of nucleophilic substitutions, right? So now you may be thinking that we all know that a catalyze that's this kind of when we have this oxygen present here which reacts faster than this and this also, but these examples are not because of the reaction center itself, okay, it is because of the neighboring group or the groups next to the carbon of the reaction center. We call this group neighboring groups. These are called the neighboring groups, okay, this groups next to the carbon of the reaction center. Mechanism by which the group speed up the reaction known as the neighboring group participation or ankymeric assistance. Now how this happens, like if there is a sulfur group present here, this sulfuric lone pair attacks here and the elimination happens. So we have this 3 member of ring intermediate, and then this this neighboring group participation of sulfide happens and alcohol, then this product is formed. So in both cases, so here also we have this participation of the oxygenic lone pair, but there is a slight difference, okay. In both cases ionization of the starting material is assisted by the lone pair of electron rich functional group. In the ether, in the first example, which is this one, is the assist by forming the pi bond, okay, but the sulfide is assisting by the formation of sigma bond. So here pi bond formation is happening by the given of this lone pair of the oxygen, but here a sigma bond formation is happening. This feature is known as the neighboring group participation and also the ankymeric assistance. And here also one more important feature is the formation of the cyclic intermediate, which commonly happens during a neighboring group participation or ankymeric assistance, okay. So now last question of today's session is, ATP is required as a cofactor to provide the necessary phosphate group, and the active site remains long open as long as the messenger molecule is bound to the dash receptor. Fill in the blanks. Number A, G protein receptor, number B, ion channel receptor, number C, kinase link receptor, and number D, ligand mediated receptor. In which case of receptor the ATP is required as a cofactor to provide the necessary phosphate group, and the active site remains long open as long as the messenger molecule is bound to the receptor. In which kind of receptor the ATP is involved? G protein receptor, ion channel receptor, kinase link receptor, and ligand mediated receptor. Let me check because we don't have much time, so I will just discuss this question. But still I have many questions, many answers I think. Number 8, I think I have number A, number B, number B, number C. Okay, let me check if number B is the correct one as mentioned. So the correct answer will be number C. So always remember, kinase are the enzymes which are associated with the ATP. Okay, kinase are the enzymes where the ATP is binds to in the active site. Okay, because this particular kinase it acts a kinase. So whenever there is ATP is mentioned, so try to find if there is a kinase mentioned also, because kinase are the enzymes which are directly related to the ATP. So basically what happens in case of the your kinase linked receptor, so there is a signal binding site here, and we have the tyrosine. So in kinase enzyme, basically tyrosine, serine, threonine, tyrosine, these are the amino acids which are involved, because these are the phenolic OH, and how we will discuss in later. Okay, in upcoming classes we will discuss about the kinase and this kinase linked receptor in more detail. So first the signal molecule, this is a signal binding site, and the signal molecule comes and joins to the signal binding site. Okay, and we have, so so first this receptor or this gate was open, right, but when there is a particular signal molecule, there we have this kind of dimer formation. Okay, and this dimer have this we have this activated relay protein which takes the cellular response, and this cellular response after this signal dimer is formed, and this dimer, when the dimer is formed, we have this ATP is transferred to the ADP, and the phosphate is one phosphate is transferred. Okay, so this is the activated tyrosine kinase regions, and this is the fully activated receptor tyrosine kinase. Okay, and after this phosphate is transferred from this ATP to this tyrosine of this particular receptor, kinase link receptors, we have this cellular response from this kinase link receptor. So ATP is required to as a cofactor in this kinase link receptor to pass their phosphate group to tyrosine, so that this particular receptor is activated and they can pass the cellular response to other proteins. Okay. So this was the last question of today's session. So today we discussed successfully about week 1 and our week 1 and week 2, right. We discussed many things about starting from the basics of medicinal chemistry, then we discussed about many things regarding the enzymes, catalysis, some about organic chemistry like anchimeric assistance, acid based, and everything. So all of you have participated very actively in today's class, which is very necessary. So this kind of class we will have every Tuesday from 6 p.m. to 8 p.m. Try to join on time. Okay, this lecture PPT and this video both I will share soon on the NPTEL website. You will find it on the SWAM portal. I am sharing my email ID in the chat box. Okay. So this is my email ID. If you have any doubt or any query, you can email me on this ID. Okay, I will happy to answer your queries. In the next class we will discuss about week 3, and I hope today's class it will help you in solving your weekly assignments and understand the course medicinal chemistry better. Anything, any query, just drop an email to this email ID which is mentioned in the chat box, and I hope all of you have understood the things I have told you. And if anything you have any doubt, just I will just let me know. I will discuss in next class. Okay, thank you all for joining. I will close the session for today. Let us meet on next Tuesday from 6 p.m. to 8 p.m. for the next week class. Okay, Thank you all. I will just close the session for today.